Hello, dear students and my dear friends. Today I am going to discuss about next RNA virus that is also a part of the human retroviruses which causes cancers in human beings. We have already in my previous topic, we have already seen about the human immunodeficiency viruses, which is also a type of retrovirus causing oncogenesis in human beings. So as in the previous topic, we have seen that the three important RNA viruses which causes oncogenesis are HIV and in hepatitis C viruses, that is a group of Flaviviridae family which causes hepatocellular carcinoma, and third again a type of retrovirus that is also called as the HTLV or human T cell lymphotropic viruses. So before proceeding to our topic, I would like to request you that my channel, please subscribe to my channel, Learn with Anshul, and please do like and share if you if you gain any knowledge from my lecture, because it will help me motivated to make more videos for you. So let's start today's video. So HTLV, that is an oncogenic RNA virus. So here you can see the structure of an HTLV virus. So before proceeding to the this in detail about the HTLV virus, I would like to discuss some of the important points regarding the retroviruses. So in the case of retroviruses, we have already seen that there are different type of retroviruses like alpha retro, beta retroviruses, and delta retrovirus, gamma retrovirus, and again here the some of the retroviruses contains simple genomes like the avian leukosis virus belonging to alpha retroviruses and murine leukemia viruses. So basically these as avian for the birds, and murine also for the rodents. So here mostly they are the animal virus oncogenic viruses. Again here we can again say that mouse memory tumor virus, that is also of the beta retroviruses. Next, simple, these all, that is the alpha retrovirus, gamma retroviruses, and beta retroviruses contains simple genome. That is, they have only structural genes, which is the most important. That is the GAG, POL, and ENV genes, which we already have discussed. That they basically make structural part of our of the virus, that is the capsid, matrix, and envelope part. In addition, and some the in the enzyme, that is the reverse transcriptase, polymerase, and integrase enzyme. Next is the viruses which contains the complex genome. In the complex genome, the first is the human immunodeficiency virus, which belongs to the group of lentivirus, and human T cell lymphotropic virus, which is a type of delta retrovirus. So human T cell lymphotropic virus is a type of delta retroviruses, and the human immunodeficiency virus belongs to the lentivirus subgroup. Now here one thing, that virus they may carry these oncogenes which are responsible for responsible for causing oncogenesis in the host cells, or else they may also not carry, and they may modulate the cellular genes in such a manner that they become convert themselves into the malignant cells. Next important point is that here we can see that most of the retroviruses causes infections in the mostly the oncogenesis is caused in the animal as a host, and only few RNA viruses causing oncogenesis in the human beings, and especially the lentivirus, that is the HIV, and HTLV belonging to the Retroviridae family, and hepatitis C virus causing hepatocellular carcinoma belonging to Flaviviridae family. So coming to the HTLV, here you can easily recognize the structure of the human T cell lymphotropic viruses. And basically, again, here, as we have discussed, that in the case of HIV, also there was two types, HIV-1 and HIV-2, in which HIV-1 was the most common type of virus, most common type, which is found in the human beings in comparison to HIV-2, which is confined to or restricted to some of the parts of the world, especially in African countries. Similarly, here also there are four basically the HTLV has been also been categorized into four types, that is HTLV 1, 2, 3 and 4 type, in which the HTLV1 and HTLV2 share almost 60 to 65% of their genetic similarity in their genetic, and these two only are responsible for causing oncogenesis in human beings, and in that also predominantly by HTLV1, whereas in the HTLV2 causing oncogenesis is rare, and HTLV3 and 4 causing oncogenesis is not still proved. So here we can see here, so it is an enveloped, like all retroviruses, it is also an enveloped virus, and the nucleocapsid is composed of capsid proteins. You can see here in the case here, this is the capsid part below the envelope protecting the nucleic acid. So here the P24, that is the protein, capsid protein, which helps in the genome protection. Then comes the in that viral genomic RNA, that is single-stranded RNA. You can easily see two single-stranded RNA plus genetic code. Then reverse transcriptase, which is synthesized by the P55. You can see here P55 is the reverse transcriptase. Then protease is denoted by the P10, that is hydrolysis of viral, responsible for the hydrolysis of viral peptides, and integrase, that is the P32, that is the help in the integration of viral DNA with the host DNA, which causes the formation of provirus. Next is the viral matrix is composed of P19 protein, and it surrounds the nucleocapsid. So matrix surrounds the nucleocapsid, and capsid surrounds the nuclear part, genome part. Then the viral envelope, which is the top, which is the upper part of the virus, is composed of a lipid bilayer plus the viral glycoprotein GP21 and GP46. So in their structure, you can say that it is almost similar to the HIV virus with some differences. Now, if I go to more specific here, specific in this part, so you can see here that the stalk part is made by GP21 and in upper part by GP46. 46. These are the envelope proteins, which is coded by ENV gene. Then comes the P10 protease, P55 reverse transcriptase, and P24 is the capsid, P32 integrase. Then you can see here the RNA, then P19 matrix, and single-stranded RNA, two strands of single-stranded RNA. So if come to the first of all to the introductory part, so this HTLV1 human T cell lymphotropic virus was firstly identified in 1980, and it belongs to the delta retrovirus gene of the genus of the Retroviridae family. Interhuman transmission of HTLV1 infection occurs most likely through breastfeeding, contaminated blood or blood products, and sexual contact. There are evidence demonstrating that exposure of the HTLV1 infected cells allows transmission of cell-free viral particles to CD4 lymphocytes and other cell types. It means that if any of the HTLV1 infected cells is exposed to the to any of the other CD4 lymphocytes, so it helps in the transmission of this HTLV1 cells or HTLV1 virus to other cells, healthy cells or other cells which are free from viral particles. However, cell-to-cell transmission and clonal proliferation of infected cells are more efficient in propagation of HTLV1 virus. So, cell-to-cell transmission and clonal proliferation in infected cells are the two important mode of propagation of HTLV1 virus. So coming to the pathogenesis, so first of all, the human T cell lymphotropic virus or human T cell leukemia virus is cancer-causing replication competent human retrovirus. The first retrovirus, which was discovered by Gallows et al., ever linked with a human cancer affecting 10 million globally. Virus has sporadic distribution of endemic areas, origin through founder effect, and exemplifies RNA virus genome and replication. It is an enveloped virus with 80 to 100 nanometer diameter of the virion, having two covalently bonded single-stranded RNA strands, each of 9 kilobase within the nucleocapsid core. The virus evades the immunes to establish any of the infection, any of the pathogen. It is necessary that it should escape the immune mechanism of the host cells, and each of the pathogen have different method of evading the immune system, because in such cases immune cells are either not able to recognize the pathogen, or they are mostly destroyed if the pathogen are very pathogenic, so they can destroy the host immune cells. So here we see that how the virus evades the immune surveillance is through a smart molecular mechanism and is latent in the infected person for a prolonged period. So it becomes inactive, or we can say that it does not show any type of multiplication or proliferation in the host cells, and it remains in a latent stage for a long period, and as the opportunity comes it converts itself into an active form. So in 2 to 5% cases at a later stage, it manifests as severe to potentially fatal to discrete type of diseases. So mostly they remain latent, but in whichever host, if it converts itself into the infectious form, it causes two types of diseases. First is the cancer of mature CD4 plus T cells, which is also called as the adult T cell leukemia or lymphoma. That is, in short, it is ATL, and a chronic inflammatory central nervous system diseases, HTLV1 associated myelopathy or tropical spastic paraparesis. So one is the disease or in disease of the T cells, mature CD4 plus, basically T helper cells, T cells, CD4 plus T cells called adult T cell leukemia or lymphoma. And the next is the a chronic inflammatory central nervous system disease, which is also called as the tropical spastic paraparesis. So transmission is mostly transmitted through infected body fluids, including breast milk, which is also called as the vertical transmission, that is mother to child, then blood by through transfusion of infected blood or accidental injury, then or accidental prick through from infected person to a healthy person, blood, then semen or cervical secretions, and organ transplant, which is organ transplant is a type of horizontal transmission. In addition, there is a lack of therapeutic regime and prevention of transmission routes. Restricted breastfeeding or formula feed with screening of potential blood and organ donors, use of condoms to prevent sexual transmission can contain the virus across the world. So these are some of the preventive measures. Surveillance for HTLV-1 based on routine reporting of new diagnosis of HTLV-1 or associated diseases or prevalence surveys. If adopted by each country, in addition, a proper registry for cancer would contain the virus across the globe. Human T-cell leukemia virus type 1 was first isolated in 1980 in a T-cell line derived from a patient with cutaneous T-cell lymphoma. Basically, it was given by Poise et al., Poise and his co-workers in the year 1980, and was shown to be the etiological agent, causative agent of adult T-cell leukemia. ATL that was given by Hinuma et al. 1981, Yoshida et al. In 1982, and Takasuki in 2005. Though these were the separate works, which was which was done by the different scientists through the world, and they isolated. They were the scientists who isolated the this HTLV from the basically from the T cells, because it is one of the important cause of causing important type of causing a mature infection in the matches. They have an affinity towards the T cells. In 1985, HTLV-1 seropositive patients in French Martinique were diagnosed with a neurodegenerative disorder termed tropical spastic paraparesis, basically, which is a neurological disorder. In short, it is also called as TSP, which was first isolated by Gessin et al. In the year 1985. A similar clinical disorder was reported in Japanese patients and was named HTLV-1 associated myelopathy, HAM. Osame et al. Worked by Osame et al. In 1986 and Osame and Igata in the year 1989. HTLV-2 is rarely pathogenic and is sporadically associated with neurological disorders. The work done by Jelly et al. In the year 1992 and Arwajo and Hall in 2004. There have been no known diseases associated with HTLV-3 or HTLV-4. Next, here, one important thing that human T-cell leukemia virus or T lymphotropic virus type 1 is a member of the Delta retrovirus family, which also includes the simian T lymphotropic virus type 1, HTLV-1, and bovine leukemia virus BLV. They are basically animal viruses, and there are 4 known strains of HTLV, that is HTLV-1, 2, 3, and 4. And HTLV-1 and HTLV-2 are prevalent worldwide, given by Edlich et al. In 2000 and Proiti et al. In 2005, while HTLV-3 and HTLV-4 have been identified only in Central Africa, Mahox and Gesai in 2009. Then HTLV1 and HTLV1 and HTLV1 are highly related. They are HTLV2 and HTLV3 are closely related to HTLV2 and HTLV3 respectively. Thus, the HTLVs are thought to derive from irrespective transmission between monkeys and humans. So it is basically it is thought, or it is not proven still, that scientists believe that it is basically through the transmission from between humans and monkeys, and the genetic variations, because they have a close genetic homology between them. That is the simian T cell lymphotropic virus and human T cell lymphotropic virus. So the genetic variations among HTLV1 and strains is less than 8%, and HTLV1 and HTLV2 show 70% nucleotide homology, proven by Gesa et al. In the year 1996 and Fewer and Green in the year 2005. So if you see here, the mechanism of replication in the case of HTLV1. So through this diagram, if you see here, some of the important component I can show you, that in this case, this virus is this infection. You can see here, this is the GLUT1 receptor present in the host cells, through which this HTLV1 gets attached, this surface unit, and here after that it is fusion of viral envelope and cytoplasmic membrane takes place. Then if I show you in the detail, if I can explain you in detail about this, I can through this figure I can show that HTLV1 mechanism of surface in cell infection and replication. Surface subunit of HTLV1 glycoproteins interact with heparan sulfate proteoglycans, that is also called as HSPGs, heparan sulfate proteoglycan. See here you can see this is the part of heparan sulfate proteoglycans. Okay, and of the targeted cells cytoplasmic membrane. A complex is then formed between the viral envelope, HSPGs, and neuropilin, that is the NRP1. Here you can see in the here is a neuropilin, HSPG, and the viral genome. So here you can see that surface subunit of HTLV1 glycoproteins interact with heparan sulfate proteoglycans of the targeted cell cytoplasmic membrane. A complex is then formed between the viral envelope, HSPG, heparan sulfate proteoglycans, and neuropilin 1 and glucose transporter 1, that is the GLUT1. You can see here, this is the GLUT1. So a complex is formed between the surface subunit HTLV1 envelope protein, then glucose transporter 1, HSPGs, and neuropilin. So here the envelope fuses with the cytoplasm. The envelope fuses with the cytoplasmic membrane, and the viral RNA is released into the cytoplasm. Okay, so here you can see that there is a fusion of viral envelope and cytoplasmic membrane of the cell, and the viral RNA is released into the host cells. So this is the mechanism of the entry of the viral RNA into the host cells. Now after that, if when they interact with the cell surface receptors and enter into the host cells and release the viral RNA, they undergo reverse transcription. And we know that this is the characteristic of the retrovirus, that is the a process of reverse transcription by the enzyme called reverse transcriptase, or we can say RNA dependent DNA polymerase. So as they are converted into cDNA. So basically this RNA does not directly synthesize protein, but first they convert themselves into complementary DNA, and then this complementary DNA undergoes replication, and again they synthesize one an additional strand, and they convert themselves into double-stranded cDNA. Okay, so this viral RNA is first converted into the DNA copy of the genome RNA, and is the within, and this formation takes place in the cytoplasm of the cell. So after they convert themselves to the produces a DNA copy of the genome RNA within the capsid in the cytoplasm, then this DNA will enter into the nucleus of the cell. And after entering in the nucleus, it will integrate with the in the it will integrate into the host cell DNA. You can see here that viral DNA enters into the, first of all, the viral DNA is formed in the cytoplasm of the cell, host cells. And after that, they enter into the nucleus of the host cells, and they again integrate with the host cell DNA at any random point. And thus, they develop or convert themselves into a proviral DNA. Okay, and they form this, thus they form as a provirus. So this is called as the provirus are those virus in which the genome of the virus get integrated with the host cell DNA. After integration, so provirus serves as a template for the development, or the serves as a template for the synthesis of viral transcripts. So after that, the next step, that is again the synthesis of messenger RNA. So this proviral DNA, first of all, it gets integrated into the host DNA, and they serve as the template for the process of transcript transcription of the viral DNA. And here some of which are unspliced, and after that they convert into the they convert into the messenger RNA, and these messenger RNA get converts into, they undergo post transcriptional modification. As we know that there are two parts in the RNA after transcription, that is exons and introns, and introns are removed and exons come together to form messenger RNA. So here it goes under post transcriptional modification, and they convert themselves into viral RNA, and they are then released from the nucleus of the host cells. And after releasing from the host cells, they get again translation. Translation is the process through which the RNA or messenger RNA converts itself into the proteins. And these RNA, when undergoes translational changes or process of translational, they convert into themselves in, they form themselves to four proteins, that is the GAG, POL, ENV, and proproteins. And after that, they undergo assembly, and they form a new viral structure. They form a core surrounding them, and after that, through the process of budding, see here basically the capsid or the nucleocapsid is formed, and when they are getting released from the host cells, that is here the virus release and maturation, they form the envelope of the virus. So they finally After releasing from the host cells, they form or they get matured and form envelop in the cell. So the integrated, so here what happens, the some of which are spliced and will serve as mRNAs. Viral proteins are synthesized. The proteins and genomic RNAs assemble and particles bud from the cell. Capsid proteins are proteolytically, so what happens that after the budding, they are getting released from the cell, and these capsid proteins are proteolytically processed by the viral protease enzyme and producing mature infectious virions. As you can see here, that this is mediated by the viral by the protease enzyme. So this is the cycle, cell cycle, or the mechanism of cell infection and replication of HTLV-1 virus. Next, here we have already discussed that here protease and structural proteins are formed, and these proteins alongside two copies of viral RNA migrate to the budding site and are released from the cell surface to further mature into infectious viral particles following protease dependent activity. Next is that retroviruses named human T cell were as isolated from patients with cutaneous T cell lymphoma and leukemia in 1980 in USA, and HTLV-1 is present worldwide and has been established as the causative agent of certain cutaneous T cell lymphomas of adults as well as the nervous system degenerative disorder causing tropical spastic paraparesis, which we have already discussed. So here one thing, interesting thing is that it does not carry an oncogene. So HTLV-1 does not carry an oncogene. So how does it is responsible for oncogenesis? So we will discuss here. First of all, we will see that the structure of the genomic structure of this RNA. So human lymphotropic virus have a marked affinity for mature T cell, and HTLV-1 is expressed at very low level in infected individuals because most of the time they remain latent in the cell and only few of them convert themselves into the infectious cycle. So HTLV-1 is expressed at very low level in infected individuals. It appears that the viral promoter enhancer sequences in the long terminal repeat. So basically, we have seen that uh, if you have gone through my previous lecture, you have seen that at 5 prime and 3 prime in there are U5, U3, 5, U3R and U5 parts. That is also called as the LTR part of the viral genome. And here they are also called as the long terminal repeats, and they contain the promoter and enhancer sequences, and they may be responsible to signals associated with the activation and proliferation of T cells. If so, the replication of virus may be linked to the replication of host cells, a strategy that would ensure efficient propagation of the virus. So here basically it is said that the LTR the region which is present in the viral genome which contains the viral promoter enhancer sequences, it may be responsible to signals associated with the activation and proliferation of T cells. So if unless and until they if it enhances the activation, this promoter and enhancer sequences in the LTR, when the this gets integrated enter inside the host cells, they suddenly start proliferating the T cells, and due to proliferation the cells start multiplying. The T cells activation and proliferation, the cells starts multiplying above their normal rates, and as such it becomes easier for the virus to get spread from one cell to host cell. As we have seen the mechanism also, that basically one of the important mechanism of transmission is through the cell propagation. So it helps it to uh, infect new cells easily because the cells start multiplying, and this is mostly the LTR region is mostly responsible. So here we can say that the replication of virus may also be linked to the replication of host cell strategy that would ensure efficient propagation of the virus. Next important thing is that the human retrovirus are trans-regulating. That means that they carry a gene which is also called a tax gene, whose product alters the expression of other viral genes. Trans-activating regulatory genes are believed to be necessary for viral replication in vivo and may contribute to oncogenesis by modulating cellular genes that regulate cell growth. So here both gene which is present, as we have seen that in the LTR, these genes are present, and they are mostly that trans-regulating, means that it can control the expression of other genes. So basically, it helps because itself it does not contain any oncogene. So it basically hold the it takes control over the mechanism of the host cell gene and start regulating. And as such, more multiplication of or more proliferation and activation of host cells and more propagation of the virus takes place. So make mechanism the HTL may cause cancer by a mechanism different from that of other retrovirus because it has no viral oncogene. Rather, it has two special genes in addition to the standard retroviral genes called TAX and REX that play a role in oncogenesis by regulating mRNA transcription and translation. The TAX protein has two activities. First, it acts on the viral long terminal repeat sequences to stimulate viral mRNA synthesis, and it induces NFKB, which stimulates the production of interleukin 2 and the IL-2 receptor. The increase in levels of IL-2 and its receptor stimulates the T cells to continue growing, thus increasing the likelihood that the cells will become malignant. And the Rex protein determines which viral mRNAs can exit the nucleus and enter the cytoplasm to be transmitted. So basically, two genes are mostly responsible for the mechanism of oncogenesis, that is the tax and rex gene, and we have seen that how it stimulates the interleukin 2 and its receptor and continuous growing of the T cells, which finally causes malignancies. So these viruses are distributed worldwide, although less than 1% of the people worldwide have HTLV1 antibody. More than 10% of the population in endemic areas will be seropositive, and antibody may be formed in 50% of relatives of virus positive leukemia patients, because we have seen that the mode of transmissions different through body secretions, blood, and other sources. So they other individuals who are in contact with the patient can also have the antibody in addition to the patient himself or herself. So next is the transmission of HTLV1 involves cell-associated viruses. Mother to child transmission via breastfeeding is an important mode. Efficiency of transmission from infected mother to child is estimated at 18 to 25%. Blood transfusions, sharing blood contaminated needles, and sexual intercourse are the effective means of transmission. Seroepidemiology has linked infection with HTLV1 to a syndrome called HTLV1 associated myelopathy and tropical spastic paraparesis. The primary clinical feature is development of progressive weakness of legs and lower body. The patient's mental faculties remain intact. HAM-TSP is described as being the same magnitude and importance in the tropics as multiple sclerosis in Western countries. Similar to multiple sclerosis, the same symptoms in the tropical spastic paraparesis patients. H2LV2 has been isolated but not been conclusively associated with a specific disease. So with this, we conclude our topic, that is the HTLV1 human retrovirus causing oncogenesis in human beings. I hope this video would have been informative, and I thank you all for your patience learning and giving your precious time for watching my video. If you do like, please like and subscribe it. Thank you.